Let's return for a minute to the myelin sheath. We talked about how the myelin sheath gets generated along the axon of certain neurons. We said that some neurons are myelinated, others are not. We said that myelin is a very fatty material and it's laid down over the surface of the axon in these discrete regions with spaces in between. We said that the myelin is produced in the peripheral nervous system by Schwann cells, and it's produced in the central nervous system by oligodendrocytes. We said that the myelin sheath is discontinuous, and that the places where there is no sheath get a name. They're called nodes, nodes of Ranvier. Now, these nodes are placed very precisely along the length of the axon, about every 1 to 2 millimeters down the length of the axon. And it's at the node that the sodium channels are very abundant, because it's at the node where new action potentials can be generated, where propagation can occur. In fact, we give this a name. We call this saltatory propagation. Saltatory just meaning sort of jumping propagation. Everywhere that the axon is myelinated, that's the area where the signal is just going to travel forward. Every time we hit an unmyelinated node of Ranvier, we're going to generate a new action potential. Send it along till we hit the next node, generate a new action potential, send it along. So myelin has a purpose. Myelin is an insulating material. And what myelin allows for is propagation of the action potential without a loss of strength. In some neurons, this is critical to their function. On this slide, we can see a myelinated axon on a neuron. Here's the cell body. Here are the dendrites coming off the cell body. Here's the axon hillock. Here's the myelin running down the axon. They're showing us the cells as the darker orange color. Remember, if we're in the periphery, these are Schwann cells. They're going to wrap themselves around the axon and secrete their myelin in layer upon layer. Here are the axon terminal branches. Signal is going to be propagated this way. It's going to be generated, travel, then regenerated, travel, regenerated, travel, regenerated. And everywhere that it travels underneath myelin, none of that strength is going to be lost because the sheath is insulating. Think about the axon like an electrical cord. The electrical cord that's attached to your television carries electricity from the wall to the television, and it travels along that cord underneath a plastic coating. Inside that television cord, there are wires. There are wires that are transmitting the electrical signal, the electrical current, I should say. And that plastic coating around those wires insulates the signal. It prevents the signal from simply traveling away from the wire into the air. It prevents the signal from being lost in strength as it moves from the wall to your television. The myelin is doing the same thing. The myelin is keeping the electrical current within the axon and not allowing it to leak out of the axon into the air, losing strength. So the whole purpose of the propagation of action potentials down the length of the axon is to get that signal somewhere. The signal was transduced into electrical current, and then it was sent along the length of the membrane, down the axon. It will travel into those terminal branches, and then it will be passed along to another cell. And remember, it depends on whether you're on the sensory side of the peripheral nervous system or the motor side, whether the signal is being sent towards the central nervous system or away from the central nervous system. So there are only certain cells that are going to be involved in this. You might have a neuron communicating with another neuron. You might have a neuron communicating with a gland, or you might have a neuron communicating with a muscle cell. What we want to talk about next is the junction between these cells, what we call the synapse. The synapse is critical because this is the place where this information, this electrical signal, has to be sent along its way to the next cell. And the synapse is that point of communication. There are two kinds of synapses. There are what we call electrical synapses, and there are what we call chemical synapses. Let's talk about electrical synapses first. Now, we've got two cells involved. For ease, we're going to talk about two neurons. We're going to talk about a pre-synaptic neuron and a post-synaptic neuron. The signal is coming down the axon of the presynaptic neuron. It's going to have to be sent across the synapse to the postsynaptic neuron. And then we needed to travel along the length of the postsynaptic neuron. Now, at an electrical synapse, those two neurons are going to be connected to each other through structures called gap junctions. Now, gap junctions, like the name suggests, are connecting points between two cells, between the membranes of two cells, that are built in a way to encourage things to pass through them, through the gap. When you look at the structure of a gap junction, it's made out of 6 individual subunits of protein. These are called connexin proteins. And the 6 of them all joined together creates what's called a connexon. So connexons in the presynaptic membrane and connexons in the postsynaptic membrane are going to join together to create a gap junction. Now, what can pass through this gap? Well, it's a pretty small gap, so only very small molecules and ions. Electrical synapses and their gap junctions provide for a way for cells to communicate with each other with virtually no delay. As you'll see in just a minute, this provides for very close communication between two cells. So we're going to find electrical synapses in parts of the nervous system where the speed is really important. The speed of transmission of signal from one cell to the next is critical. A good example of this is in cardiac cells. In order for the heart to pump in a regular, predictable way, the electrical signal that gets generated has to be transmitted from cardiac cell to cardiac cell to cardiac cell in a very predictable, very rapid way. And so we find a lot of these electrical synapses in the heart. On this slide, we're looking at a drawing of electrical synapse. So in this drawing, you can see this structure right here that widens out. This is one of the terminal branches of the axon. And this is a widened area at the bottom that we sometimes call a knob, a synaptic knob. So the cell body would be way up here with its dendrites. Then the axon would come down, and then at the end of the axon, it would split into terminal branches. Here's one of those terminal branches, and it's ending right here at this widened area called a knob. So here coming down the length is the nerve impulse. Now they're showing it inside of this axon terminal, but remember it's really traveling down the membrane. Here is the postsynaptic neuron. So what you're looking at here is the membrane of a dendrite. Terminal axon, dendrite. The two membranes are coming together and they're actually being joined with gap junctions. They're being connected to each other with gap junctions. We're going to take one of these and we're going to blow it up. Here's presynaptic membrane. You can see the heads in dark yellow, tails in light yellow, heads in dark yellow. Here's the postsynaptic membrane. Here's the synapse, the space between them. Notice that it's a pretty narrow space. They're telling us it's only 20 nanometers across. That's not even the distance of one microtubule across. Now, the areas in purple, these are connexons. And each connexon has 6 individual connexin proteins inside of it that are joined together to create this structure. They've cut the connexon in half here, so we can see that there's a tunnel in the middle of it, a gap. The connexons in this presynaptic membrane are going to join together with connexons in this postsynaptic membrane, and they are essentially creating a tunnel. So signal comes down this membrane, goes through the tunnel, and comes into this membrane. And we have transmission, very rapid transmission. These two membranes are only 3.5 nanometers apart from each other, where the gap junctions are. That's a very, very small distance between those two membranes. We're going to find electrical synapses in areas where the speed of transmission from the presynaptic to the postsynaptic is critical. Now, chemical synapses are very different. For ease of discussion, again, we're going to talk about a synapse that involves 2 neurons. So we've got a presynaptic neuron and a postsynaptic neuron, and there's going to be a space between them. There's going to be a synapse where they communicate with each other, but there's going to be a much wider space. Now we're talking about 20 to 50 nanometers between these two membranes. And we're going to give the space a name. We're going to call it the synaptic cleft. The action potential that arrives at a chemical synapse has to be transduced. Whereas in an electrical synapse, the electrical signal is going to pass through as an electrical signal. In a chemical synapse, the electrical signal is going to be converted into a chemical signal. It's the chemical that's going to cross the synapse, cross the cleft. And then when it hits the postsynaptic membrane, it's going to be converted back into an electrical signal. So you can imagine that transmission through a chemical synapse is slower because we have this transduction event that has to happen. Now, the chemicals that are involved in a chemical synapse are called neurotransmitters. Neurotransmitters are a fascinating group of chemicals. They're produced up in the cell body of the neuron. They're packaged into transport vesicles, and then they're sent down the length of the axon. Remember, through fast axonal transport. Kinesins are going to carry them down microtubule tracts, down into these terminal branches, into these bulbs at the end of the terminal branches, what some people call synaptic knobs at the end of the branches. The vesicles full of neurotransmitter get stored right there down at the end of the terminal branch in these knobs or bulbs. So the transduction event happens when the action potential travels down the membrane of the axon, and we're going to see the vesicles containing the neurotransmitter move forward and fuse with the presynaptic membrane, similar to what we talked about for exocytosis. The vesicles are going to fuse with the membrane, the presynaptic membrane, and they're going to release the neurotransmitter out into the synaptic cleft. Now, that neurotransmitter will pass across the cleft and it will hit the postsynaptic membrane. It's a chemical, and chemicals bind to receptors. Neurotransmitter is going to bind to receptors on the postsynaptic membrane. And this interaction between the neurotransmitter and the receptor is going to do one of two things. It's either going to help generate an action potential on that postsynaptic membrane, or it's going to inhibit action potentials from being generated in that next neuron. So the advantage that a chemical synapse has over an electrical synapse is a variety of responses. The electrical synapse is just sending signal very quickly from one neuron to the next neuron. The chemical synapse can play with that signal a little bit and either send it along or alter it, inhibit it from happening. Now, if we have a stimulation event happening on the postsynaptic membrane, we're going to call that excitatory postsynaptic potential. Remember, when we're talking about changes in charge along the length of a membrane, we're talking about membrane potential, we're talking about electrical potential. And in the case of an excitatory postsynaptic potential, we're going to see depolarization happen on the postsynaptic membrane. Sodium channels are going to open, sodium's going to rush in, we're going to start a new action potential. But we can also see inhibitory postsynaptic potential on the postsynaptic membrane, and that's achieved through hyperpolarization. On this slide, over on the left, you're looking at a drawing of a chemical synapse. Here's a terminal branch of an axon and the bulb at the end, or the knob as it's sometimes called. Here's the space, the synaptic cleft. Here's the postsynaptic membrane. So this would be dendrite on the next neuron. Again, they're showing us electrical current coming down. They're showing it in the cytoplasm, but remember, it's really traveling down the membrane. Down here, we have little vesicles, as you can see, little vesicles, and they're containing neurotransmitter. Notice too that mitochondria have been sent down here. Mitochondria have been sent down to live in this bulb. Now, they've taken this little area of the drawing and they've blown it up for us right here. What has to happen to send the signal from this neuron to this neuron across this chemical synapse is that these vesicles full of neurotransmitter have to fuse with the membrane, and we need exocytosis to occur. The neurotransmitter is going to travel across the synaptic cleft and it's going to bind to these postsynaptic membrane receptors. In some instances, that binding event is going to regenerate action potential. Those are considered EPSP, excitatory postsynaptic potential. Or the binding event will cause an IPSP, inhibitory postsynaptic potential. Up at the top of the right-hand side, you're looking at an actual chemical synapse under an electron microscope. So let me see if I can orient us. This is the membrane of an axon terminal right here. Over here, this is the membrane of a dendrite on another neuron. These are all little vesicles full of neurotransmitter. We call them synaptic vesicles. They've got to fuse with this membrane, release their neurotransmitter into the synaptic cleft, where it can bind to the postsynaptic membrane and cause something to happen, either an excitatory potential or an inhibitory potential. So it all depends upon what receptor is present on the postsynaptic membrane. The neurotransmitter is going to bind to one or the other kind of receptor, depending on the neuron. We give these two different types of receptors names. One of them is called ligand-gated or ionotropic. The other one is called metabotropic. Now, when I talk about these two kinds of receptors, I'm not talking about one being for EPSPs and one being for IPSPs. I'm talking instead about one of them having a direct action on the postsynaptic cell and one of them having an indirect action on the postsynaptic cell. Let's look at the ionotropic ones first. Ionotropic neurotransmitter receptors are simply ion channels. So when the neurotransmitter binds to an ionotropic receptor, it's going to cause that receptor to change shape and open up. It's an ion channel. Binding of the neurotransmitter opens it up. Ions can pass through, either into or out of the postsynaptic cell. So an ionotropic receptor, a direct acting neurotransmitter receptor, can either stimulate or inhibit postsynaptic action potentials. It depends on what kind of ion channels it acts as. Some ionotropic receptors act like sodium channels. Some ionotropic receptors act like potassium channels. If it acts like a sodium channel, it's going to depolarize the postsynaptic membrane. It's going to create an EPSP. If it acts like a potassium channel, it's going to hyperpolarize the postsynaptic neuron. It's going to act as an IPSP. Now, metabotropic receptors are different. They are indirect acting. So they don't act like actual ion channels. When the neurotransmitter binds to a metabotropic receptor, what happens is it activates a second messenger. And it's the second messenger that then opens up a nearby ion channel and the ions can pass through. Again, a metabotropic receptor can generate either EPSPs or IPSPs. It depends on the cell. But there's an extra step involved with a metabotropic receptor. The neurotransmitter binds to the receptor, sets in motion a second messenger, which opens up an ion channel nearby, which sends ions through. So a metabotropic receptor is just a little bit slower than an ionotropic receptor. On this slide, we're looking at those two types of neurotransmitter receptors. So these would be found in the membrane in the area of dendrites on a neuron. This image on the left represents an ionotropic type of receptor, a direct-acting neurotransmitter receptor. And this represents a metabotropic receptor, or an indirect-acting receptor. So in this drawing, this area right here would be the synaptic cleft. The presynaptic membrane, the presynaptic cell would be up here. The neurotransmitter would get released into the cleft. They're showing it to us as this blue circle. When that neurotransmitter binds to a direct-acting ionotropic receptor, it's going to cause that receptor to change shape. The receptor is going to act just like an ion channel. It is an ion channel. The shape change is going to be opening it up. If it's a sodium ion channel, sodium's going to rush in. We're going to see depolarization occur. A new action potential will get generated in this second cell membrane. We're going to have an excitatory postsynaptic potential. If it's a potassium channel, when the neurotransmitter binds, we're going to see the opposite. We're going to see potassium leave the cell. The inside is going to get hyperpolarized. It's going to be hard to generate an action potential here. We're going to make it harder to generate an action potential. Over here, the neurotransmitter is shown in pink. It's not going to bind to an ion channel receptor. It's going to bind to a different type, a metabotropic receptor. And when it binds, it's going to cause that receptor to change shape. That shape change is going to make something else happen. It's going to activate some kind of second messenger. And that second messenger is then going to bind to and open up a nearby ion channel. So there are two classes of neurotransmitter receptor. There are ionotropic, direct acting, and metabotropic, indirect acting.